Switching dreams on and off? That and more in Science for this week. A team of researchers from Heriot-Watt University are the first to successfully 3D print stem cells using valve-based techniques. This amazing feat was achieved by overcoming the challenge of printing the extremely sensitive live cultures without damaging them. The hardware has been made to be delicate enough to handle and 3D print induced pluripotent stem cells while keeping the biological functions intact. These kinds of technology are huge steps forward for the 3D printing of complex organs. Harvard University researchers have developed a metamaterial that allows any light passing through it to go infinitely fast. This on-chip metamaterial is made by embedding silicon pillars in polymer wrapped in gold film. This is found to exhibit a refractive index of 0. The material is hailed as an evolution in fiber optics and could lead to photonic-based telecommunications over modern electronic versions. This will ultimately allow researchers to manipulate light at the nanoscale without the risk of energy loss. Neuroscientists from the University of Berkeley, California, have been able to control when a sleeping mouse enters a dream by simply flipping a switch. This has been achieved by inserting an optogenetic switch into the medulla and activating the neurons with a laser light. Activating the neurons would then send the sleeping mouse into REM sleep, which is the state at which one experiences dreams. When the neurons were kept from activating, it completely hindered the mouse's ability to go into REM sleep. Researchers from Newcastle University and the Science and Technology Facilities Council have produced the first synthetic model of a bacterial outer membrane. This amazing breakthrough gives researchers an incredibly valuable tool in the development of more effective antibiotics. The structure of the particular bacterial membrane was determined with a polarized neutron reflectometer. This instrument allows for extremely precise measurements of the film layers at the atomic level. Scientists are harnessing the distinct abilities of viruses to transfer energy, and they're using quantum mechanics to do it. In a combined effort involving many different biologists, engineers, and quantum physicists from MIT, researchers have manipulated a virus into mimicking a plant cell. This is achieved by placing a virus in contact with synthetic dyes that could mimic photosynthesis. This enables the virus to learn the plant's actions, such as delivering energy. A hole about the size of 50 Earths has been found on the surface of the Sun and has been spewing solar winds in our direction. The hole, which is located on the corona, the outermost layer of the Sun, is in the northern hemisphere. Holes like this are often the source of faster than normal solar winds. These regions have a lower density of solar material and are generally a lower temperature. These factors in combination make it easier for solar winds to escape, and this particular one has been sending solar winds right in the direction of Earth. The last few weeks, several auroras have been seen on Earth as a direct effect. Science has been after the secrets of aging since the days of alchemy. So far, it's been especially difficult to see how aging affects neurons, since the subjects would need to remain alive for the experiments to be reliable. Mice and worms have taken the place of human subjects for these reasons, but there are still quite a few differences in genetics when you compare a mouse to a human. To try to solve this problem, scientists have begun using induced pluripotent stem cells from patients' skin and coaxing them into becoming neurons. However, a problem that remained was that the conversion process turns back the biological clock, making the derived neurons appear as younger cells regardless of their original age. By skipping the induced embryonic stage, they found this could also be solved and the gene expression profiles could remain intact. Huntington's disease is a neurodegenerative genetic disorder where a mutant protein called huntingtin is produced that damages and ultimately kills brain cells. Aside from disruption of muscle coordination, mental decline is often present, which leads to dementia. A new experimental class of medicine that involves gene silencing has been used in the latest clinical trials. In order to create the mutant huntingtin, the corresponding messenger RNA is synthesized from DNA. The RNA is then translated for protein synthesis. The gene silencers are a mirror copy of the RNA, binding to it and marking it for destruction, which prevents protein synthesis. The drug is injected into the spinal cord once a month for 4 months. Let's hope this amazing breakthrough saves a lot of lives. That was FTW. Thank you for liking and commenting. See you next week.